Many of you know that my lab works on innate immunity, and from that angle, we rediscovered pyroptotic cell death that is mediated by the Gasdermin family of pore-forming protein. So we're getting into this topic by studying antibacterial innate immunity, and we believe that this kind of pro-inflammatory cell death may also promote antitumor immunity, and that's the focus what I want to cover in my presentation. So I'll start by showing you what is pyroptotic cell death, pyroptosis. And in this movie, you will see the cells undergo a lytic death. You see the cells are cell body are swelling with bubbles blowing out, and eventually the dying cells undergo burst with membrane lysis, and that will release cytosolic contents and trigger a strong pro-inflammatory response. And this form of programmed cell death, which basically is a necrotic cell death, is very different from what we know about apoptosis. And you can see in apoptotic cell death, you see the cells did not swell, but instead shrink with plasma membrane integrity maintained. And at the same time, the dying cells also have phosphatidylserine flipping from the inner leaflet to the outer leaflet. And that serves as the eat-me signal for macrophages to engulf the dying apoptotic cells. So pure apoptotic cell death will not trigger inflammation, which is very different from pyroptotic cell death, which is highly pro-inflammatory with swelling membrane rupture. So pyroptotic death was originally observed in 1986 in anthrax lethal toxin treated macrophages. In 1992, Arturo Zeglinski also observed a cell death, macrophage cell death in response to Shigella flexneri infection or bacterial infection. And so for many years, we misregarded this cell death as a special form of apoptosis, but indeed, and this cell death is highly pro-inflammatory, serve as a host defense response, particularly in response to bacterial infection. In 1996, Zeglinski showed that this pyroptosis require a caspase, a specific caspase called caspase-1. So caspase-1 is downstream of this so-called inflammasome pathway, which is one of, aside from C-gas sting pathway, the inflammasome pathway is another major cytosolic innate immune defense pathway, not only against bacterial infection, but also defending against many types of danger signals in the cytosol. Many of you have heard this NRP3 molecule that is can detect some danger signals. So the canonical inflammasome in response to bacterial infection, for example, recognizing bacterial flagellin or sensing some bacterial toxin activity, and then recruit pro-caspase-1. Pro-caspase-1 becomes activated to process interleukin-1 beta as well as interleukin-18. And that indeed is how caspase-1, as the first member of the caspase family, discovered as an enzyme to process interleukin-1 beta. So active caspase-1 also trigger pyroptotic cell death and induce immune activation. And for many years, and we have no idea how does active caspase-1 stimulate pyroptosis. So in 2015, my lab and Vishwa Dixit's lab independently identified this molecule called Gasdermin-D or GSDMD. And this protein is absolutely required for caspase-1 to induce pyroptosis. And actually is also required for secretion, not processing, but secretion of interleukin-1 beta and interleukin-18. And 1 year later, my lab and Hao's lab, Judy Lippmann's lab, and we showed that this protein is a substrate for caspase-1, and the cleavage will liberate the N-terminal domain that can make pores on the plasma membrane. And Hao's lab further determined the cryo-EM structure of a single of the Gasdermin pore. As you can see, this beta barrier structure will be inserted into the plasma membrane that is responsible for the the membrane rupture during pyroptosis. And for each pore, you have about 27 to 28 protomers. So it's a very large pore that is about 20 nanometer in inner diameter. So it's much larger than the nanopore you have heard this morning. So, so with that, we basically figure out the mechanism on downstream of caspase-1 in inducing this pro-inflammatory lytic death that is done by the N-terminal domain of Gasdermin-D when it gets cleaved by caspase-1. So in addition to this so-called caspase-1 mediated canonical inflammasome pathway, we have another pathway called a non-canonical inflammasome. And so work from my lab and also Visual Dix's lab basically establishes this pathway. So in this pathway, we have another caspase in mice called caspase 11, in human called caspase 4 and 5, and they were bypass the requirement of a null-like receptor, but instead directly recognize lipopolysaccharide in the cytosol. So there is a physical interaction between LPS and the CAR domain of caspase 11 or caspase 4-5, and this will activate the caspase, and the active caspase 11 4-5 will also cleave after this FLTD motif in Gasdermin-D, and then liberate the N-terminal pore-forming domain. You make pores. So in the next about 15 minutes, I would basically touch on 3 points. And the first point is the LPS activation, or cytosolic LPS activation of non-canonical inflammation or pyroptosis in antibacterial defense and sepsis. And I will talk about unpublished work which reveals an immune activation mechanism upon caspase 4 or Gasdermin-D induced pyroptosis. And I'll end by summarizing our work on Gasdermin-mediated pyroptosis in triggering antitumor immunity. So in bacterial infection, so we took advantage of 2 bacterial infection models, Bacterial thailandensis and Shigella fractionarii. And both bacteria are intracellular free living, which means that they will inevitably expose their LPS to be recognized by caspase 4-11, and then you need will stimulate Gasdermin-D dependent on pyroptosis. So in a mouse model of bacterial infection, like a Bacterial thailandensis infection, we do see a very striking phenotype or protective effect of the caspase 11 Gasdermin-D pathway, because wild-type mice can survive the infection, but the knockout mice, caspase 11 or Gasdermin-D knockout mice succumb to the infection. However, for a very similar bacteria called Shigella fractionarii, we found there is no phenotype. So if you delete caspase 11 or Gasdermin-D in the mice, so wild-type mice, we don't see a protective effect. Wild-type mice behave similar as the knockout mice. And the reason for that is because the bacteria has evolved an inhibitor protein called RSPC3, and this toxin from Shigella fractionarii can directly target the caspase 4-11 and inhibit its activation. And if we remove RSPC3 from the bacteria, now we can see that the wild-type mice can mount the defense response and survive the infection, while the knockout mice succumb to the mutant infection. And consistent with that, activation of pyroptosis will limit the bacterial replication in the infected liver and the spleen. Mechanistically, and this protein RSPC3 uses NAD as a donor to post-translation modify a specific arginine in caspase 4-11, and this modification has never been discovered before, so we give a name as ADP riboxynation. So this bulky ADP riboxynation completely inactivate the caspase, and so that the bacteria can shut down this immune defense pathway. So if we compare mutant bacterial infection and wild-type bacterial infection in mice, we clearly found that the mutant bacterial infection can allow the mice to generate a higher level of antibody against IgG antibody against the Shigella fractionarii. So that's part of the antibacterial protection mechanism. And this was not seen in caspase 11 Gasdermin-D knockout mice. So we can combine this RSPC3 mutation with 2 other life-attenuated bacterial mutant, and we can see a much higher antibody production. So if we secondly challenge the mice with lethal amount of, or lethal dose of Shigella fractionaria, we can see a vaccine-like protective effect once the mice can activate the innate immune pyroptosis pathway. So this data suggests that activation of pyroptosis in the infected mice or bacteria-infected mice can activate humoral immunity and stimulate antibody response. So for this pathway, caspase 4-511 recognize LPS, and upstream of that, there's a family of innate immune protein called the GBPs that is interferon gamma-induced GTP binding protein, and those proteins can attack intracellular bacteria and cause cell wall stress and promote the release of LPS as well as other bacterial molecules from the bacteria so that it could be detected by the innate immune system. So indeed, bacteria, as an evolutionary arm race, as you heard from James's talk, so the bacteria has evolved various kind of mechanism to hijack this pathway. So back in 2017, and we showed that Shigella fractionaria has evolved a ubiquitin ligase, IPH 9.8, can ubiquitinate the GBPs, and it causes its proteasomal degradation. And I just show you that RSPC3 from the same bacteria can target caspase 4-511 and catalyze this novel post-translation modification to shut down the pyroptotic pathway. And work from Vishwa Dixit's lab also found that IPH 7.8, another ubiquitin ligase bacterial protein, can directly ubiquitinate Gasdermin-D and it causes the proteasomal degradation. So the bacteria has evolved multiple mechanisms to hijack this pathway to ensure the shutdown of this important antibacterial innate immune defense pathway. So this really highlights that this pathway is critical for antibacterial defense. So the evolution has already done the experiment. However, if the pathway goes excessively, and you will get a septic shock with gram-negative bacterial infection. For example, if you inject high dose of LPS into the mice, you get endotoxic shock. The mice die within 1 day. The knockout wild-type mice, the caspase 11 or Gasdermin-D knockout mice, they can survive very well. So this suggests that when you have pyroptosis occurring everywhere in the body, and that will cause this so-called septic shock. So in this septic shock or sepsis, there is a clinical phenomenon called sepsis-associated encephalopathy, which means that the recovered patients may have brain issues. They become retarded or other brain issues. And this is due to the disruption of blood-brain barrier during sepsis or during a high level of infection. So here is, many of you know, the structure of the BBB. You have brain vascular endothelial cells lining up, and outside you have pericytes and astrocytes. So we can model this infection-induced BBB disruption in the mice. So you can see that in this mouse model of septic shock, using Evans blue as a tracer for the integrity of BBB, of blood-brain barrier, you can see that in the septic shock mice, and the BBB was completely disrupted, a very, very strong phenotype. And if we do that in the caspase 11 and Gasdermin-D knockout mice, we see a complete protection from LPS or infection-induced BBB breakdown. So we also characterize the BBB structure, ultra-structural changes using electron microscopy. You see those black cells are dying and brain endothelial cells. And those cells, they were detached from the basal membrane. You see the enlarged space here. And as a result, that the junction between the endothelial cells will be disrupted. So that's why you get BBB disruption. So we using fluorescence labeling approach, we also found that Gasdermin-D is highly expressed in CD71 positive brain vascular endothelial cells, but not neurons. So Gasdermin-D is only expressed in microglia cells and brain vascular endothelial cells, but not in the neurons. So we can use this AAV specific targeting brain endothelial cells to rescue the phenotype, which I don't have time to show you the data. in this BR1 AAV that is specific for the brain endothelial cells. We can show that the Y-type Gasdermin-D can rescue the blockage of BBB disruption, but the caspase 11 resistant, cleavage resistant mutant cannot, suggesting that the disruption of the BBB indeed is due to brain endothelial cells sensing of LPS and undergo pyroptosis or undergo pore formation in the plasma membrane. So we also use another Cree line that is specific for the brain vascular endothelial cells. So using this specific Cree line, we can delete Gasdermin-D specifically in brain vascular endothelial cells, and we can show that the deletion of Gasdermin-D from those cells can completely block the BBB disruption induced by LPS or dual septic shock. So the whole inflammasome pathway, you heard caspase 1 and RP3, and those so-called canonical inflammasome pathway, and the whole history can be traced to IL-1 beta. And IL-1 beta is a cytokine mainly expressed in monocytes and macrophages. So we profiled the expression profile of inflammasome components in about 100 different cell lines derived from non-macrophages. And you can see that the canonical inflammasome pathway, caspase 1, pro-IL-1 beta, they are rarely expressed in those non-macrophage cells, consistent with what is being known in the literature or in the field for many years, because the canonical inflammasome pathway mainly operates in macrophages. However, if the non-canonical pathway, caspase 4, Gasdermin-D, are widely expressed in those non-macrophage cells, and it actually correlates very well. And those cells are mainly from epithelial tissues, And we also found that IL-18 is highly expressed in those cells and show a strong correlation with caspase 4 and Gasdermin-D. So this raises an interesting hypothesis that we think maybe IL-18 is a cytokine downstream of caspase 4/5, the pathway. So because their expression profile highly correlate. So we took those cells that are positive for caspase 4, Gasdermin-D, and IL-18 expression, and we transfect LPS into the cytosol. We can clearly see robust pyroptosis. We can also see IL-18 secretion in the supernatant. And if we do Salmonella typhimurium infection, we got the similar results. So those cells, as I showed you in the earlier slides, they can sense cytosolic LPS, they can sense bacterial infection, and they induce pyroptosis and IL-18 production. So under in vitro conditions, we use activated caspase 4 to process pro-IL-18. We can see very efficient processing of pro-IL-18 into the mature form. And the processing or the cleavage efficiency is even higher than caspase 1, because caspase 1 is the only enzyme known to be able to process interleukin 18. So, however, most importantly, the mouse protein caspase 11, although it can also bind LPS and become activated and cleave Gasdermin-D, but activated caspase 11 barely cleave pro-IL-18 in the mice. So basically, the pathway doesn't work in the mouse system, only works in the human system. So this suggests that the caspase 1 pathway in macrophages, downstream of the inflammasome pathway, is not the dominant pathway for IL-18 production. Instead, it is the LPS-activated caspase 4/5 in epithelial cells. That is the predominant pathway for IL-18 production. So many immunologists actually know that IL-18 is everywhere, not only just in macrophages. So, and we established pro-IL-18 as a cytokine target for caspase 4/5 sensing of LPS, cytosolic LPS, and this links pyroptosis to type 1 immunity. And we are now generalizing a humanized mice to reconstitute the pathway in mice so that we can revisit the immunological function for IL-18 in the future. So we are in collaboration with Dr. Jingjing Ding's lab at the Institute of Biophysics at Chinese Academy of Science, and we determined a crystal structure of active caspase 4 in complex with pro-IL-18. So the active caspase 4 is a dimer. The dimer has this little interdimer small beta sheet, and this small beta sheet binds to specifically recognize pro-IL-18. So it's a 2-2 enzyme substrate recognition. So and this recognition actually is very similar to caspase 4 recognition of Gasdermin-D, which we published 3 years ago. So in both cases, the active caspase using this exosite rather than the catalytic pocket, then use this exosite to recruit, specifically recognize and recruit Gasdermin-D as well as pro-IL-18. So it's very similar mechanism. And the structure also explains why caspase 11 doesn't work. Because in the caspase 11 structure, and this L4 loop flips towards the exosite, towards this region, and creates a steric clash so that the caspase 11 cannot bind to pro-IL-18. So we can make a mutation, make a 3 residue mutation here to regain the activity of caspase 11 to process a pro-IL-18. And this structure is also the first time to allow us to see what the pro-cytokine looks like. So you can see that this is a pro-cytokine, pro-IL-18 structure, and the pro-peptide folds back, have a very integrated into the main body, and the cleavage of the to remove the pro-peptide and triggers the dramatic conformation rearrangement. You can see here. So there's almost a refolding here. So it's a conformation maturation of mature IL-18. So that is important for people who are interested in engineering cytokine to make like a cytokine drug. So we not only have Gasdermin-D, and in human we have ABCDE is a family of protein. It's a family of protein. They all contain this 2-domain architecture, N-terminal pore-forming domain, normally inhibited by the C-terminal inhibitory domain. And so they all have intrinsic pore-forming activity. That the difference is that they are activated by different signals, which I will show you a bit later on. And as you heard from James Chen's talk, that this family of protein is also highly conserved in lower organisms, including bacteria. So the work from Philip Kranzushi's lab showed that some bacteria have gasdermins, that their gasdermins are quite active, and they can make even larger pores upon cleavage by a protease encoded in the same operon. And this pore pathway is also important for bacteria to mount antiphage defense. So once the bacteria sense some phage infection, and the protease becomes activated by some phage stimulated signal and the cleave bacterial gasdermin, and then you get cell death. So the single bacteria dies to prevent the spread of the phage and protect the whole entire community. And the work from Louis Glass' lab in Berkeley showed that in fungi you also have bacterial gasdermin-like proteins. So in fungi you have this so-called allorecognition phenomenon. 2 haploid strands fuse together to become a diploid. If the 2 haploids are genetically incompatible, and the fused diploids will activate the fungal gasdermin and the cells, the misfused diploid cells will die out of fungal gasdermin activation, pore formation. So in both cases, you can see that in bacteria and fungi, the pathway activation of the pore-forming activity or gasdermin-like pore-forming activity serve as a defense function, either antiphage defense or genetic defense. So, and back to a few years ago, we actually have been quite actively working on in this area. And many years ago, we showed that Gasdermin-E, which is originally known as a deafness gene, is activated cleaved by caspase 3, and that will convert the apoptosis to pyroptosis, because caspase 3 is very well known to induce apoptosis. However, in cells that are positive for Gasdermin-E expression, and active caspase 3 indeed would switch the cell death to pyroptosis, and that actually brings a lot of functional implications in different biological process, because caspase 3 is activated in many different cellular processes. And I'll show you that, I'll summarize the work we did on Gasdermin-B. So what we found is that Gasdermin-B is transcription upregulated by the interferon gamma signaling. And then Gasdermin-D is Gasdermin-B is cleaved by granzyme A from cytotoxic lymphocytes, including CD8 positive T cells or NK cells. The cleavage after this lysine residue will liberate the N-terminal domain and then make pores. And this granzyme A is from cytotoxic lymphocytes, and we show that this pathway actually it works in antitumor immunity. In this CD26 syngeneic mouse model of colon cancer, we can show that this granzyme A Gasdermin-B pathway has a synergistic effect with the checkpoint blockade in this mouse model of mouse syngeneic model. So this data suggests that Gasdermin-mediated pyroptosis could be represents an effective mechanism in lymphocyte killing or anti-tumor immunity. So, however, if we artificially deliver active Gasdermin into tumor cells in the mouse model, without the T cells, we just use the nanoparticle, as you heard from previous talk, with a controlled release manner. So we conjugate active Gasdermin to nanoparticles. We also have a small molecule to control the release of Gasdermin from nanoparticles. And the system is a bioorthogonal that can target specifically, selectively to tumor cells in the mouse model. So this is a collaboration work with Dr. Zhibo Liu's lab. So with this approach, we can kill about 10 to 15 tumor cells in the mice. And we found that killing of 10 to 15 tumor cells by pyroptosis is sufficient to cause complete tumor shrinkage, and in the wild-type mice. But in the T-cell deficient mice, we did not see this phenomenon, suggesting that this little bit of tumor cell pyroptosis is remodulate the tumor microenvironment and stimulate tumor immunity. Indeed, we can show that CD4, CD8 cells, and we profile all those immune cells, and we can use antibody to neutralize this tumor growth inhibitory effect. And from all those data, we conclude that pyroptotic cell death in the tumors can convert cold tumor to hot tumor. So I'll end to acknowledge the people in the lab who did all this work, and I'll stop here. Thank you.